peripheral nervous system is to the axon. But if that damage is to the cell body of that neuron is dead. So therefore, in the peripheral nervous system only, if you damage the axon of those neurons, then potentially there can be some form of regeneration. Okay, but that regeneration takes a long period of time, as if, let's say, you slice your finger off and your finger is like dangling down and stuff like that, and you run to the ER and they reattach your finger, you will lose that function for a certain amount of time before the neuron reestablishes the connections, right, within the muscles itself. So therefore, in the peripheral nervous system, once you damage the axons, immediately white blood cells from the blood is going to invade that area and start to clean up all of those cellular debris. It's going to phagocytose, eat up all of that cellular debris, and also destroy or eat up all of the axons from the point of damage all the way to the axon terminal. Okay, so it's going to get rid of all of that axon. So now that effector is no longer stimulated by that neuron. So if that's a skeletal muscle, that skeletal muscle will become paralyzed, right? Which means it cannot become activated without that neuron. And that neuron now is truncated, so it has a portion of its axon just now gone. Okay, so it has a stump now. Now, the second thing that happens is that weeks after the injury, once all the cleaning up has already occurred, the Schwann cells come together and they fuse together to form a long regeneration tube. That regeneration tube starts from the damaged portion of the axon and reestablishes connection with the original muscle that is controlled by that neuron. Okay? And then from within that regeneration tube, growth factors are released. So the growth factors can be released by that neuron and also by the surrounding cells to initiate the regrowth of that axon. So the axon starts to slowly regrow until it reestablishes connection with that muscle once again. Once the axon reestablishes connection with that effector, then the regeneration tube will dissipate. All the Schwann cells basically will disengage from each other, and new Schwann cells will come in and reestablish myelin sheath around those particular axons.
This is for adults?
This is for adults.
Okay.
Yes.
What would cause an axon to damage?
So if you, let's say you're chopping things and you accidentally slice your finger off, that axon's gone, right? So yeah. Or let's say even if something happens where you're playing around and let's say you get a laceration across your arm or whatever, and it's deep enough to cut off some of the nerve, so therefore the axons are damaged by that point. And yes, this occurs in both children and adults. Obviously, in children it's faster in terms of the healing process. In adults, it takes a longer time in order to heal. All right, so imagine here's a site of injury of this neuron. Okay. All of these white blood cells come in and start to eat up all of these damaged portion and basically eat up everything else from that portion all the way to the axon terminal, leaving this muscle now denervated. So there's no neural innervation to that. That muscle is paralyzed basically, right? You see that during this time, the cell body of this neuron is larger because it's actively making new proteins in preparation for repair. Okay. Now, at some point, a bunch of Schwann cells come together to form this regeneration tube, which reestablishes connection to this skeletal muscle, let's just say. And then growth factors will stimulate the regrowth of the axon. Now, the reason why we need this regeneration tube is so that we can have a guide to know where that axon needs to regrow to. Otherwise, the axon is just going to regrow and branch out in multiple ways and stimulates whatever you don't need to stimulate, right? So that regeneration tube is very important for that purpose. Once you reestablish this connection of that axon to this muscle, the regeneration tube will basically disappear. New Schwann cells come in and reestablish new myelin sheets around that new axon. So therefore now you regain that particular function. Keep in mind that anytime you damage anything within the body, even if you repair it effectively, you will never regain 100% of the original function. There's always some degree of loss that ensues. All right, so that's the peripheral nervous system. Now, in the central nervous system, the reason why we cannot regenerate any axonal damage is due to the fact that once you damage the axon, immediately there are these particular receptor proteins that are expressed on the oligodendrocyte called the death receptors. So death receptors automatically popped up on the oligodendrocytes, and as the oligodendrocyte binds to certain kind of signals, they go through apoptosis. So only in that immediate area, right? So the oligodendrocyte basically now goes to programmed cell death. They start dying off, which means there's nothing to form your regeneration tube at that point. And furthermore, there's also other inhibitory proteins that are secreted by all of those damaged tissue that prevents the formation of new myelin sheaths, even if there's surviving oligodendrocytes in the surrounding area, okay? So already there's a prevention or inhibition of any kind of regeneration or formation of any kind of regeneration tube, okay? Thirdly, the astrocytes from the surrounding area, once you clean up all of that debris, instead of forming a regeneration tube, which you are already inhibited from doing so, the astrocyte starts to invade those areas that is now empty. And as the astrocyte invade those areas, they start to secrete these extracellular materials we call the glial scars. So basically just scar tissue that overlays what neural tissue used to be, right? So because of those those glial scars, the neurons can't regenerate, or the axon can't regenerate. So therefore, that neuron is just gonna die. Because there's no the establishment of connection whatsoever.
I didn't fully understand why.
The why? Because of why pattern. There's no why. If the neurons in the central nervous system dies, the reason why it can't regenerate is because you have so many inhibitory factors that prevents the regeneration of those neurons. If you want to ask why, you can't ask me. I didn't make up this rule.
Oh.
I'm just telling you the rule as to why you cannot regenerate, not the reason as to why you can regenerate.
It's interesting, like why the CNS
Yeah,
they prevent,
exactly.
the most important part.
Yeah, I don't know. Maybe it's just, space saving type of activities. Yeah.
Wait, so the death receptors form on the oligodendrocytes?
Mhm.
Okay. And those are the oligodendrocytes that were attached to the cut you're on?
To those axons in the first place.
Okay.
Right. So remember, in the peripheral nervous system, once those axons are damaged, once you clean up that debris, right, one cells comes in and form that regeneration tube to guide the regeneration of the axons.
Mm.
So to prevent that from happening inside the central nervous system, all of these oligodendrocytes in that area, and of course, especially the one that surrounds the damaged axon, they also die. So once they die, there's no regeneration tube that is formed. And essentially, there is no regeneration tube that can be formed within the central nervous system, okay, no matter what. So because of all of that inhibitory proteins that are secreted out as well during that damage. So yeah, there's a lot of factors that actually prevents or inhibit the regeneration of these axons. Now, of course, through stem cell research, we can actually find different ways to stimulate sort of like the regeneration, right? But that is not a normal occurrence. So that's more in the process of whatever it is that they're trying to work out. But it's a controversial type of research because the only way you can get stem cells to regenerate neural tissue is from the zygote, which is a fertilized egg. Right? So therefore, you have to stick a needle in there, suction out some of those cells, and then potentially causes irreversible damage to this zygote. So the zygote can become an embryo and a fetus and a child, but then that child may have certain type of anatomical defect that can result from that process. So, or that zygote is still going to die altogether. So therefore, it's controversial in that sense. All right. Questions? Now we're getting into the physiology of these guys. Okay, so far everything is just anatomy. Just purely anatomy, just review for the most part, and hopefully it is reviewed. If not, that's okay. We're just introduced to it. All right, now, every single cell of your body, when they are at rest, they exist in what we call a polarized state, which means across the membrane of every living cell. So here's the outside, and here's the inside. Across this membrane, there is a separation of charges. And those charges are imposed by the concentration of ions that exist outside versus inside of the cell, and other particular factors as well. So therefore, for the most part, all living cells on the immediate environment outside of that membrane, it will always be more positive. Okay, due to a higher collection of positively charged ions congregating outside of the cell. The immediate environment inside of the cell, right at the cell membrane, we're not talking about the whole cytoplasm, but just right at the cell membrane inside, is always going to be more negative. Make sense? So in a living cell at rest, it exists in a polarized state across the membrane. The neuron is no different. So therefore, the neuron, because it is a living cell at rest, it exists at a polarized state. Now, this polarized state is created and maintained by what we call an electrochemical gradient. Electrogradient refers to the difference in charges that exist across the membrane. Okay, so more positive on the outside, more negative on the inside. The chemical gradient is due to the chemical or the ions that is distributed across this membrane, an unequal concentration. So, another way to put it, there's a lot more sodium ions outside of the cell. So sodium ions outside is very high. Sodium ions inside of the cell is very low. Okay. The potassium ions inside of the cell is very high. The potassium ions outside of the cell is quite low. Now, there's other ions involved. Let's say for calcium ions, the amount of calcium ions outside of the cell is very high. The amount of calcium ions inside of the cell is low. So this is a clear concentration difference. So this represents what we call the chemical gradient that exists across the membrane. Therefore, allowing for the outside of the membrane to be more positive and the inside of the membrane to be more negative. Okay, so existing in this polarized state produced by that electrochemical gradient. So therefore, if we take an oscilloscope, some kind of instrument, to measure the difference in the voltage across this membrane. So let's say if you take an oscilloscope, right, this particular machine has this meter that basically has like, some kind of needle that goes back and forth. You take a probe and you place it on the outside of this membrane. You take another probe and you place it on the inside of this membrane. You zero out the outside, so basically the outside voltage is 0 millivolts. And you measure the difference in that. So the inside of the membrane is always going to be some negative voltage compared to the outside, because the inside is always more negative, okay? So let's say in the neuron, the difference is about 70 millivolts. So on the inside of the membrane of this neuron, it is 70 millivolts more negative than the outside of that neuron. Does that make sense? Okay. So anytime you measure the membrane and you measure the electrical differences of the membrane, we call that the membrane potential. Okay? And the membrane potential, once again, is just that polarized state that exists in a resting cell due to the separation of the electrochemical gradient. So electrochemical gradient, charges being different, chemical gradient, concentration of these ions being different as well. Make sense? All right. So for these particular type of neurons, let's just say, because they are the one that utilizes this electrochemical gradient to their advantage to produce that massive electrical signal which they use to communicate with each other and with other tissue, they can now change their charge state by changing the permeability of this membrane to these ions. So once the membrane changes its permeability, which means it will allow for these ions to move across the membrane, then once the ion starts to move, it will also change the polarized state of that membrane. Okay? So in that case, when you stimulate the neuron can change its membrane potential, and therefore allowing for this particular neuron to basically produce any kind of electrical signals that it wants to. Alright, so when the neuron is at rest, and we measure the membrane potential of a resting neuron. I told you guys already, there's a difference of about 70 millivolts and more negative on the inside, okay? Which means the inside of this membrane of this neuron is 70 millivolts more negative than the outside. So we call this the resting membrane potential of a neuron. And the resting membrane potential of a neuron is registered, is measured at negative 70 millivolts. Okay? Precisely because we use this method to measure the difference in that charge separation. Okay? Now, there are three factors that will help the neuron maintain this negative 70 millivolts difference between the outside and the inside. The first factor is that on the inside of these cells, you have a bunch of proteins, right? Integral proteins, peripheral proteins, whatever have you. You also have a bunch of like proteins within the cell itself, right at the membrane itself. Most of those proteins will have things like phosphate attached to it. Phosphate has a lot of negative charges associated with it. And because the phosphate are associated with those proteins, we call them the large fixed anions inside the cell. Anions are negatively charged ions. And because these are phosphate that has a bunch of negative charges, and they're bound to proteins, so they're bound to these large fixed structures on the membrane, right? Fixed, that means they're not moving anywhere. And because they have a bunch of negative charges, they also contribute to these overall negativity inside of the cell. Does that make sense? Okay, all right, so that's the first factor. The second factor is that you have this very important sodium-potassium ATPase. Remember the sodium-potassium ATPase that exists across the membrane? So imagine, if you will, you have this particular sodium-potassium ATPase. So here's the outside. Once again, here's the inside, positive out here, negative in here, right? This particular sodium-potassium ATPase utilizes ATP and hydrolyzes ATP directly to become ADP plus inorganic phosphate, right? And using the energy release from this phosphate bond breakage to take 3 sodium ions from inside the cell and move it out. And take 2 potassium ions from outside the cell and moves it in. Okay? What kind of transport mechanism is this? I know we didn't have this lecture. If you guys studied it well, you should be able to answer this question. So, in terms of transport mechanism, ask yourself, is this passive or active?
Secondary active.
Now, if it's active, is it primary active or secondary active?
Secondary.
Okay, so primary active utilizes ATP as an energy source directly. Secondary active uses another ion and allow that ion to diffuse, and through that diffusion process, utilizing what we call diffusion energy to power or move any other substance against its concentration gradient. So, or power something else to move. So basically, if you remember the electron transport chain, with hydrogen ion diffuses back in through the ion channel, that diffusion powers the ATP synthase. That's almost like a secondary active phenomenon, not really a transport, but that's secondary, right? In this case, the sodium-potassium ATPase is a primary active transport mechanism. Utilizing ATP directly, it is moving 3 sodium ions against its concentration gradient because it's moving sodium ions from low to high area across this membrane. And also taking 2 potassium ions and moving these 2 potassium ions from low to high concentration across this membrane. Therefore, both sodium and potassium ions are moving against their respective concentration gradient. So if you're moving anything against their concentration gradient using ATP directly as an energy source, we call that primary active transport. Okay? This is by far the most important primary active transporter that we have in the body. If you poison this guy, game over. If this thing gets poisoned, you don't even have to worry about physiology, because you'll be dead. Okay, so don't poison that sodium potassium ATPase, okay, because this is the primary source that powers everything inside the cell. Now, you think about this, when you are moving 3 sodium ions out and 2 potassium ions in, what is the net effect in relation to the charges? Just do simple math. Positive 3 out, positive 2 back in. What's the net effect? It's always going to be a -1 inside that cell all the time. Does that make sense? So therefore, because this thing works constantly, it is automatically generating a very negative environment inside of the cell at all times. Does that make sense? All right, good. So that's the second factor that maintains that negativity inside of these particular neurons, okay? The third factor is that on the surface of these particular neurons, you also have these ion channels, and these ion channels are basically open all the time. Out and in. Now these ion channels are specific for potassium ions, but they are a type of ion channel we call the leak channels. And this leak channel is specific for potassium ions, so we call them a potassium ion leak channels. Now remember, the term leak means that when it's open and the movement of this ion is moving across this membrane, is moving in a very slow fashion. Imagine if you have a leak, it's just drip, drip, drip. It's annoying as hell, right? But it's not a lot of water coming out of that particular faucet. So therefore, this is going to allow for potassium ion to move across this membrane. In which direction is potassium ion going to move?
Inside.
You sure? So if this particular ion channel allows for potassium ion to move, you're telling me that potassium ion will move from low concentration to high concentration without energy? Right. Potassium ion then will slowly leak out of the cell, moving from inside to outside, because it's now moving in a passive movement from high to low concentration of potassium ion. If potassium ion is slowly leaving these cells, what's going to happen to the charged state inside of the cell?
Negative.
Why? Because it's losing positively charged ions. So those 3 factors is going to help maintain a very negative state inside of the cell. Right? Make sense? Okay. So therefore, if I ask you, what are the factors that help maintain the resting membrane potential of a neuron? Those are the factors. All right, that's the oscilloscope. That's basically the same thing I drew out for you guys. Okay, so now, once the membrane is at rest, you can actually stimulate this neuron to change the permeability of this membrane. So therefore, if you change the permeability and all of a sudden the inside of the cell become more positive, we call this process depolarization. So imagine, let's say if I'm just charting this particular neurons, right? So this is voltage over here. Okay, so imagine if this is the resting membrane potential of a neuron. Okay, so resting membrane potential of a neuron, let's say it's at -70 millivolts. Okay, if something happens, if I stimulate it, and all of a sudden the membrane potential changes and becomes positive, we call this, as the membrane becomes progressively more positive, we call this depolarization. Okay? Now, after depolarization, you need that membrane to go back to its resting state. So therefore, to regain its resting state, it has to become negative once again. So the negative effect, or the polarized change that allows for this membrane to become negative after it has depolarized is referred to as repolarization. Okay? Yep.
So 2 questions. Is repolarization, or R and P, is that constant or does it?
The resting membrane potential,
Yeah.
constant.
Okay.
Yeah, pretty much.
So, and then what would cause it to?
Well, I'll talk to you guys about that in a second. But right now, I'm just introducing terminology, so therefore we can apply these terms later on, once you start to change the permeability of that membrane to allow for the inside to become more positive or to become more negative, right? So we'll talk about those. So therefore, let's say this, if the cell membrane is at rest and you do something to change the permeability of that membrane, and all of a sudden the inside of this cell become more positive, then we call that depolarization. Okay, so here you can basically measure the membrane potential changes. From -70, it goes to -69, -68, -67, and so on and so forth, right? So becoming progressively more positive on the inside, okay? Now, after it has depolarized, you need to reset this membrane potential back down to its resting state. So therefore, something has to happen for this membrane to change its polarity, so it changes the permeability of this membrane to allow for the inside to become more negative once again. So that is known as repolarization. Now, once you're at the resting membrane potential, if you stimulate that particular neuron and for some reason you change the membrane permeability and the inside of the cell becomes more negative than its resting membrane potential, so basically it becomes even more negative than its resting membrane potential, we call this hyperpolarization. Okay? Make sense? Okay. All right, so how do you induce depolarization? How do you cause this polarized change or membrane potential change in this neuron? Well, let's say if you change the permeability of this membrane to allow for either sodium ions or calcium ions to move into the cell, then the inside of the cell will all of a sudden become positive. Does that make sense? Because the inside was very negative, now you're adding positive charges into the cell, right? So immediately the inside of the cell will become positive. So if the inside of the cell becomes positive, then this potential, the membrane, the voltage difference will become more and more positive. The inside here will become more positive. Make sense? So therefore, as you measure it, it will depolarize. Okay? The membrane will depolarize. That's a polarized change. That's a membrane potential change due to the movement of ions across the membrane. Okay? So therefore, if you have sodium ions and or calcium ions entering the cell due to, let's say, changing the permeability of this particular membrane, then the inside of the cell become more positive. That will result in depolarization. Okay? For repolarization, typically it's due to the membrane potential, or the membrane here becomes permeable to potassium ions. So potassium ions will now leave the cell, and as potassium ions leave the cell, the inside of the cell will become more negative again. And that typically will happen after you have depolarized that membrane. Okay? So depolarization of the membrane is typically due to the influx or the inward movement of positive charges, whereas repolarization is due to the outward movement of positive charges by changing the membrane permeability to those ions. Okay? And then hyperpolarization, if from its resting state, which is at -70 millivolts, all of a sudden you change the membrane permeability and cause the inside of the cell to become even more negative than this state. -71, -72, -73, and so on and so forth. This is due to either positively charged ions leaving the cell or negatively charged ions entering the cell. Okay? So for the positively charged ions that can leave the cell, that's potassium ions. For the negatively charged ions that can enter the cell to induce hyperpolarization, that's chloride ions. Okay? So therefore, if you change the permeability of this membrane that allow for any of those charged particles to leave or enter the cell in the appropriate way, all you have to do is just apply the math that will either add or take away, or whatever it is, to these particular charges that's already in the cell. okay? It's not that hard. If you guys think of it as hard, it will be hard. It's not. If you can add 1 plus 1, 1 plus 2, 1 plus 3, 1 minus 1, 1 minus 2, 1 minus 3, you can actually visualize some of this stuff happening. Just think of this to the nth degree, right? So instead of 1 positively charged ion entering, you have 1,000,000 of sodium ions entering the cell. You have 1,000,000 of potassium ions leaving the cell. So therefore, it's gonna cause a change in that membrane potential very rapidly. Okay. Oh, another thing to think about is anytime you have depolarization of a neuron, this equals excitation of that neuron. So when the neuron depolarizes, it becomes excited. And when it repolarizes, it just resets itself to its resting membrane potential. But when the neuron hyperpolarizes, it then becomes inhibited. And we'll see why that is in a second. All right, now for the type of neurons, or the type of ions that move across this membrane, it's gonna be either potassium ions, or sodium ions, or calcium ions, or chloride ions for the most part, okay? When they move across the membrane, remember, charged particles can only move across the membrane through facilitated diffusion, which means they have to have dedicated ion channels specific for those ions to allow them to move across the membrane. Why can't they move across the membrane without some kind of integral protein?
Because they're charged.
Yeah, because they're charged. So as soon as they enter the middle of that membrane, they get kicked back out by the non-polar tails of that phospholipid bilayer. Make sense? So they have to have a channel to allow them to move across the membrane easily. Okay? So therefore, for potassium ions specifically, you will have potassium lead channels, and you will have another type of potassium ion channel we call voltage-gated potassium channels. Now, this gets into very specific type of nomenclature or naming of these ion channels. So therefore, when you answer these questions, please write down the correct ion channels. If you say that, if you know that potassium ion is moving across this membrane in some way, shape, or form, well, I will ask you, what type of ion channels facilitate their movement? And if you say potassium channels, you will be incorrect. Because which potassium ion channels are we talking about? And they are very different for different type of activities and different changes that occurs within this changes in the membrane potential of the cell. Okay? So once we get into these discussions, please keep in mind the different types of ion channels so that you can answer those questions correctly. Right, for sodium ions, the movement across the membrane mainly is going to be facilitated through these ion channels called the voltage-gated sodium channels. You have other type of sodium ion channels too that is, less efficacious in their activities. Now we'll get to those particular ones later. All right, for the voltage-gated sodium channels, since they're called voltage-gated, they will open or close based on the changes of the voltage of this membrane. Okay? So the voltage-gated sodium channels, they will open only when this membrane reaches a voltage of -55 millivolts. That is the key to opening the voltage-gated sodium channels. The voltage-gated sodium channels also, once the membrane reaches a voltage of +30 millivolts, the voltage-gated sodium channels will become inactive. Okay? They don't close, they just become inactive. And then once the membrane reaches back down to -70 millivolts once again, the voltage-gated sodium channels will close. Yes?
So these are all voltages on the inside.
Now, voltage-gated integral proteins.
No, I mean at the values. These are the voltages
Of course,
inside.
the voltage is difference between the outside versus the inside.
Oh.
Does that make sense? So yes, measuring the difference, so always taking the outside as a 0 reference, and then whatever changes occurs on the inside. And that's how we measure. Yeah, good. Make sense? All right, so for the voltage-gated potassium channels, they will open when the membrane potential reaches +30 millivolts. And they will close when the membrane potential reaches all the way down to -85 millivolts. Okay? So just kind of keep those voltages in mind as we talk about the next activities. So here's the voltage-gated sodium channel. They have 3 states that they exist in: a closed state, an open state, and an inactive state. And they have to cycle through these states in this order. They cannot go out of order. So they are closed, and after that, they can be open. Once they're open, they will become inactive. Once they become inactive, you have to close them again before you reopen them. Does that make sense? And these are the key voltage changes that allows them to basically cycle between the closed state versus the open state versus the inactive state. Okay? So just keep those voltages in mind. And in the inactive form, you can see that it is still open. But there's a physical blockage. There's a protein here on the bottom of this particular ion channel. So when I was learning this in medical school, I had to use different methods to learn it. So I apply a real life scenario, right? So once upon a time I lived in Georgia, and I was there for about 6 years or so. And when I was there, my neighbor next to me was, a very pretty girl, right? So I would always come over her house and study, okay? And she lives on the 2nd floor though. So it's not like I can just waltz in through the front door and say, hey, how you doing, sir? And then walk up to the 2nd floor and then study with her. So I basically have to climb up the 2nd floor and enter the window to study with her, right? So then I think back like, oh, okay, that's a pretty good scenario. So I'm picturing myself as sodium ions. And I'm picturing her window as a voltage-gated sodium ion channel. So if that voltage-gated sodium channel is closed, I can't get into her room and I can't depolarize, study that situation, right? So if I'm sodium ion, if I'm entering that room, I can depolarize that room, okay? So now, if I'm looking up the 2nd floor window, her window is closed, means the voltage-gated sodium channel is closed, I can't get into that room. I have to wait until she opens the window. So as I climb up, the window's open. Now I can enter and I can depolarize that cell. I can depolarize whatever that situation is. One day, I'm climbing up the window. I see her window is open. I'm like getting excited, because I'm about to depolarize and study with her and stuff like that. And as I get up there, I see her dad standing there. And her dad is not a small dude. I mean, he's like 5'9", but he's like 285 pounds. Right? He's a big linebacker looking, guy. And he's like, son. And I was like, sir. I just went down back home. Right? So therefore, because he's standing here, he's physically blocking my entrance into a room, rendering me inactive, in a sense. So that's how I use this particular scenario to kind of learn these particular activities of the voltage-gated sodium channel. Fun time. All right, any questions so far? That's just the introduction to it, okay? Let's talk about the synapse. Now the synapse is a connection between the neurons. So therefore, this connection is referred to as a chemical connection. So therefore, it's called a chemical synapse. So when 2 neurons come close to each other, they don't physically come in contact with each other, but they come in close proximity, and there's a space between them. And the space between them we call a synaptic cleft. So the formation, the communication between 2 neurons is referred to as a synapse. Okay? So in order for one neuron to communicate with another neuron, since they don't physically come in contact with each other, one neuron will release a signal we call neurotransmitters, which will bind to the receptors on the other neuron to stimulate the activities of that other neuron. Okay? And that's how they communicate within that synapse. Okay? So imagine this is an axon terminal here. This picture is basically between an axon terminal of a neuron and the membrane of a skeletal muscle cell, but it's the same thing, same concept. So here's a cleft, there's a space between those 2 structures. Inside the cell, there's a bunch of vesicles, and these vesicles stores a lot of those neurotransmitters. So therefore, this axon terminal, this neuron, has to release the neurotransmitters into this cleft to bind to the receptors on this cell to stimulate that cell to do what it needs to do. Okay? So that's what we call a chemical synapse. Now, in terms of the connection between the neurons, if the connection between the neuron is established, then there's a synapse that occurs. So therefore, we call the neuron that comes before the synapse the presynaptic neuron. We refer to the neuron that comes after the synapse, the postsynaptic neuron. That's just a generic term that refers to any two neurons that forms a synapse together, okay? So, in that particular case, you have pre and postsynaptic neuron. Now, the way they form synapses is either between an axon terminal of a presynaptic neuron and the dendrites of a postsynaptic neuron. If that's a synapse, then we call that an axodendritic synapse. Make sense? Okay, synapse between an axon terminal of a presynaptic neuron and the dendrites of a postsynaptic neuron. Now, if the synapse occurs between the axon terminal of a presynaptic neuron and the cell body of a postsynaptic neuron, we call this an axosomatic synapse. Okay, because the soma refers to the cell body of that neuron. Now, there's other type of synapses that occurs between the axon terminal of a presynaptic neuron and an axon terminal of a postsynaptic neuron. In this particular case, we call this an axonic synapse. Okay, so there's three potential synapses that are formed between pre and postsynaptic neurons. The most common is the axodendritic synapse. The least common is the axonic synapse. All right, this is how we basically stimulate the release of neurotransmitters from these guys. So I'm gonna erase this real quick. Okay, so imagine here's the axon terminal. This is what it looks like, right? This is the terminal bouton, right, that we talked about. So axon terminal of presynaptic neuron. Okay, and then over here you have the membrane. Let's say this is the dendrite. So we'll call this dendrite of postsynaptic neuron. Okay, so this would be an axodendritic synapse. Make sense? All right. Now, here's a gap, here's a cleft. So we call this the synaptic cleft. So it's just an extracellular space that exists between these two neurons. Okay, so in this neuron, there's a bunch of vesicles. And these vesicles, and these vesicles stores or houses a bunch of neurotransmitters. So we have a bunch of these neurotransmitters inside. So these red dots equals what we call neurotransmitters. Okay? So these are signaling compounds released by the neurons, hence neurotransmitters. So they're sitting there. Now, as they're sitting there, they're basically docked, or they're bound to what we call snare proteins. So, snare proteins. Okay? And the snare proteins are attached to the membrane of this axon terminal. Nothing is happening because these vesicles are just sitting there waiting for something to happen. Well, in order for you to release the neurotransmitter, the snare protein has to allow for this vesicle to fuse with this membrane. Okay? In order for that to happen, there is a voltage-gated ion channel located on the membrane of this axon terminal, and we call this voltage-gated calcium channels. So there's a voltage-gated calcium channel located here. Now this voltage-gated calcium channel will open when an action potential, or that main electrical signal, propagates down and stimulates it. Once it opens, it will allow for calcium ion to enter the cell. Okay? Because calcium ions are highly concentrated outside of the cell, and therefore once this ion channel opens, it will allow for calcium ions to enter via facilitated diffusion, going from high to low concentration. Once calcium ion enters, it will combine with a protein, which you guys don't need to know the name of that protein. So calcium combines with that protein to form a complex. that complex will activate the snare protein. Okay? Once the snare protein gets activated, the snare protein starts to twist around each other and allow for this vesicle to fuse with the membrane of the axon terminal. So this is fusion that occurs. Okay? Once the vesicle fuses with the cell membrane, the contents inside that vesicle are released out into the synaptic cleft. Make sense? What kind of transport mechanism is this? It's called exocytosis. It's a part of bulk transport, right? So you're taking a huge quantity of material and you are transporting it out of the cell using vesicle. So in this particular bulk or vesicular transport, this is to take out any material from inside the cell, right? So now these particular neurotransmitters are secreted out, exocytosed out into the synaptic cleft, okay? Now once they reach the synaptic cleft, they will move across this cleft to reach this side of the postsynaptic neuron. Okay, so far let's just kind of cycle through this. That's just a picture, the same picture that I kind of drew out for you guys in terms of how the neurotransmitters is triggered to be released into that synaptic cleft. Please remember that, because we will go over that again, over again. Okay, and you will have to know it for this exam, for the next exam, and for the final exam. All right, now, once these neurotransmitters are secreted into this cleft, that's another terminology that we use for this neurotransmitters, for anything that is secreted out by a cell. And that term is known as a ligand. So neurotransmitters are also referred to as ligands. Anything that is produced by a cell and secreted out by that cell to potentially bind to a receptor on any other cell to stimulate that cell is referred to as a ligand. It's just a generic term for that, okay? So all of these neurotransmitters are now referred to as ligands. Now, on the membrane of this postsynaptic neuron, on, let's say the membrane of this dendrites, are a bunch of integral proteins. In this case, these integral proteins are specific for different types of ligands. Okay. So because if they're specific for different types of ligands, we call them ligand-gated receptors, which means these receptors are also ion channels that will open or close based on the binding of a correct ligand to those receptors. So hence, ligand-gated receptors. So imagine all of these are ligand-gated type of receptors located throughout the dendrites of these postsynaptic neurons. So imagine all along the dendrites and also along the cell body of this neuron, you have hundreds of thousands of these ligand-gated receptors for different types of ligands, okay? Now, there's other type of receptors we call the G protein couple receptor as well, but we'll talk about those later. So oftentimes the ligand-gated receptors can also be referred to as the ligand-gated channels because they are ion channels built into them. All right. So let's first talk about the first portion of the action potential, and we call this the graded potential. And this is generated in these postsynaptic neuron once they receive the activities of these ligands. So just imagine, right, for any of these ligand-gated receptors or channels, they are specific for a ligand. So let's say if there's a ligand that will bind to this ligand-gated receptor, and it just so happens that this ligand-gated receptor is specific for sodium ions. So therefore, once the ligand binds, these receptors will open, sodium ions will move into the cell. Make sense? Because it's moving from high to low concentration via this facilitated diffusion process. Now imagine if you have another type of ligand-gated receptor and it will bind to a different ligand, right? So imagine these are just different ligands than the first one. Once that ligand binds to these ligand-gated receptors or channels, these guys open. And it just so happens that this type of ion channel is specific for calcium ions. So the calcium ions will also enter this cell. When sodium or calcium enters the cell, what's going to happen to the charge state inside of the cell? Will it become more positive or will it become more negative?
More
It will
positive.
become more positive. And when the membrane becomes more positive from its resting state, what do we call that?
Depolarization.
Depolarization. So when sodium ions and calcium ions enter this postsynaptic neuron, it will produce a depolarization effect. Okay? So this portion of the membrane becomes positive from the inside. Just this area, not the whole thing, just this immediate area. Okay, so these particular electrical signals that are being produced, very, very tiny electrical signals that are being produced throughout these areas as long as they have these particular type of ligand-gated receptors. Now, remember I told you guys, depolarization equals excitation. Okay, so therefore these tiny electrical signals that are produced by the inward movement of sodium ions and or calcium ions through these ligand-gated receptors are collectively known as the excitatory postsynaptic potentials. Okay? Because depolarization equals excitation. So therefore, in this very small region, so imagine this is happening right here, you will produce that excitatory signal. Okay? So the excitatory postsynaptic potentials, we call them the EPSP. Okay? You want to abbreviate this, make sure you abbreviate it correctly. If you want to write it out, I love it, you guys can write it out correctly as well. Okay? So you have EPSPs being generated throughout the dendrites and cell body of this postsynaptic neuron. I'm assuming this is the postsynaptic neuron, okay? All right, now for these other ion channels here, so imagine if the ligands bind to these specific ones here, and these guys open. Okay? And when they open, they are specific for chloride ions. So chloride ions will enter this cell because there's more chloride outside of the cell versus inside of the cell, okay? And then there's another one of these ligand-gated receptors. Once the ligands bind to these guys, they will also open. And when they open, they allow for potassium ions, because they're specific for potassium ions, they allow for potassium ions to leave the cell. Because potassium is highly concentrated inside versus outside. So the movement of all of these ions are passive movements via what we call facilitated diffusion. Okay? So what happens when chloride ions enter the cell or potassium ions leaving the cell? What's going to happen to the charge state inside of the cell? Will it become more positive or will it become more negative? It will become more negative, right? So we talk about the terms that when the membrane of a cell is at rest and something happens that causes the membrane to become even more negative, we call that hyperpolarization. So these two ions moving across this membrane is going to produce hyperpolarization inside the cell. And we also mentioned the fact that hyperpolarization is the same thing as inhibition to this neurons. So therefore, these tiny electrical signals will produce what we call inhibitory postsynaptic potentials. Okay? Or what we call IPSPs. So imagine throughout the dendrites and cell body of this neuron, there's a series of EPSPs and IPSPs generated. It all depends on the type of ligand-gated receptor that is located in those areas of this neuron. And it also depends on the type of ligands that are secreted by the neurons. Okay? So there's a series of EPSPs and IPSPs generated massively around the dendrites and cell bodies of this neuron. And both of those, or all of these, are added together. So all the EPSPs and IPSPs are summated together. So in this process, we call summation. And the summation event results in what we call a graded potential. Okay. So at the beginning of this neuron and the activities of this guy, so imagine you have a neuron, and this is, we're measuring the membrane potential changes that occurs within this particular cell. If the cell's at rest, we call this the resting membrane potential. If you stimulate it, let's say you release these ligands, and the ligands bind to all of these ligand-gated receptors, and it causes either the generation of EPSPs and or IPSPs, and you take all of that signal and you add them all together. So you summate them all together, and you get a net effect. And that net effect is referred to as a graded potential. So in this particular case, the graded potential kind of looks like this. So it's the result is a very slow depolarization of the membrane. Now, if you see this, what can you say about the amount of EPSPs compared to the amount of IPSPs that you generated in that neuron? Will you have equal amount, or will you have more EPSPs compared to IPSPs?
More
have more EPSPs compared to IPSPs. What happens when you have the same amount? Then it becomes flat. What happens when you have more IPSPs compared to EPSPs? Hyper. Okay, because remember, you're measuring this from rest. Okay, so that's the graded potential.
So when you have a situation when at the apex of the graph,
Up here?
yeah,
Okay.
so what's happening there? Is it?
We haven't gotten there yet.
Okay.
We will. Not yet. Maybe not today. But I'm pretty sure you guys are quite saturated with all of this right now. Okay? So let's stop here. Then we'll go to lab. And then I'll explain any further.